Hi, my name is Eric Gonzalez,
I'm Taran Hunter.
I'm Brynn Lowry.
I'm Ted Spinola.
and this is our plant physiology video project. In order to accurately model and predict the degree to which climate change will affect the diverse ecosystems of California and the rest of the world, it is imperative to understand the drought-adapted life history strategies of various plants. Our study focuses on 3 diverse ecosystems, all located within the UCSC Upper Campus area. We collected water potentials and stomatal conductance for a total of 7 species from 3 distinct plots to measure the degree to which traits involved in water transport and transpiration vary between these 3 habitats. We predict that plants from these diverse habitats will have adapted differing strategies for dealing with their unique climates and water regime. The first ecosystem we will be studying is a riparian forest. The riparian forest is categorized by its highly dynamic dependence on fresh water. We expect these species to have the fewest drought resistance adaptations, as they have the easiest access to water. The 3 species we surveyed in this habitat are the sword fern, tan oak, and redwood. The next ecosystem is the redwood forest, an incredibly important ecosystem for California and dependent on a high water regime of coastal areas. We expect these species to maintain high transpiration rates, even with drought, and experience very low leaf water potentials. The 3 species we surveyed in this habitat are the redwood again, Douglas fir, and live oak. Our final ecosystem, the chaparral, is defined by Mediterranean summers, low nutrient, shallow, and rocky soils. We expect these species to exhibit isohydry, the drought avoidance method, and rely on readily closing stomata when they experience a drop in leaf water potential. The 3 species we surveyed in this habitat are the manzanita, madrone, and live oak again. In order to calculate the water potential for the species, we would need to set one of their leaves into the pressure chamber. Not all trees have the same amount of light exposure, so to standardize the water potential, we placed a dark plastic bag over a segment of their branches for 15 minutes. Then we would remove the bag, cut a leaf from it, set up the pressure bomb, place the leaf or stem inside with a tiny bit sticking out, then we would slowly increase the gas pressure until water would be visible in the stem, and record the pressure in the chamber. These are our results from the pressure bomb experiment. And here is the average water potential for each habitat. To find the stomatal densities of the samples, we transported the cut leaves and branches back to the lab to look at a section of each leaf under a microscope and count the stomata in a set region. To do this, we first had to coat the abaxial side of each leaf in nail polish and let them sit for a couple of minutes. Once dried, we peeled off the layer of nail polish, and with it, the top layer of the abaxial leaf cells. Using ImageJ and AmScope programs, with the microscope zoomed in to 40 times magnification, we were able to get accurate reading of stomatal density in a given area. We then recorded the stomatal densities of each species. These are our results. And these are the average stomatal densities for each habitat. As waves of drought continue to hit California, various habitats are differentially affected. Our results suggest that the habitats that will be hit the hardest by drought and potentially have the least resilience when it comes to dealing with prolonged drought exposure are those habitats consisting of species with lower water potentials over long durations. As climate warms, droughts are expected to become more frequent as well as more severe.